Now we will discuss about genetics and molecular techniques. First we will discuss about karyotyping. So karyotyping is basically study of chromosomes for abnormalities. It is always related to chromosomes. Its purpose is study chromosomes for abnormalities. So it has two processes. First, first process we use colchicine. There are two processes of karyotyping. One is for colchicine is used, and in second process chromosomes are stained. First, colchicine is added. It disrupts mitotic spindles, and then that leads to arrest cells in the mitosis process. In the second process, chromosomes are stained, ordered, and numbered by morphology, that is shape, size, arm length ratio, bending pattern. So these are ordered in three ways: morphology, size, arm length ratio, and bending pattern. So karyotyping is basically used in blood, bone marrow, amniotic fluid, and placental tissue. So for these samples we use basically karyotyping to study chromosomes abnormalities. Clinical use of karyotyping are: it detects chromosomal imbalance, for example autosomal trisomies, sex chromosomes disorders. So these are clinical uses of karyotyping. Its main purpose is karyotyping is always related to chromosomes to study chromosomes for abnormalities. So we have discussed about. Now we will study about fluorescence in situ hybridization or FISH. We will discuss about its principle, uses, and findings. Fluorescent DNA RNA probe binds to a specific gene or chromosome site. So basically it is used to study genes and chromosomal anomalies. So basically fluorescence in situ hybridization is used to localize genes and visualize chromosomal anomalies. Findings of fluorescence in situ hybridization: microdeletion, translocation, and duplication. In microdeletion, no fluorescence at locus compared to a normal copy. Translocation, abnormal signal due to chromosome exchange. Duplication, extra copy, trisomy, tetrasomy. So we have discussed about fluorescent in situ hybridization. It's basically use a fluorescent or that is stained DNA or RNA probe to study specific gene or chromosomes. So it localizes genes and visualize chromosomal anomalies. So findings: microdeletion. If no fluorescence at locus compared to a normal copy, it means there is microdeletion of gene. Translocation, abnormal signal due to chromosome exchange. And duplication, extra copy. If there is extra copy, then there is some trisomy or tetrasomy. We'll discuss about molecular cloning. Definition of molecular cloning is production of recombinant DNA in bacterial or eukaryotic host. It's basically a process of production of a recombinant DNA that takes place in bacterial or any other eukaryotic host. So applications of molecular cloning are large scale production of human proteins. So these proteins are very important overall. So these proteins are insulin and growth hormones. So insulin is, we all know that it's used basically for treatment of diabetes related conditions, and growth hormone. So these are applications of molecular cloning. Now we will discuss about gene expression modifications. Transgenic strategies in the mice: random insertion, targeted insertions, or deletion. In random insertion, gene is inserted anywhere, constitutive expressions always active. Targeted insertions or deletion via homologous recombination. Knockout, gene is removed. In knock-in, gene is inserted. It allows conditional expression that is controlled. So we have discussed about transgenic strategies that are basically used in the mice. So there are two processes: randomized or random insertion of gene and targeted insertion or deletion of via homologous recombination. So this process is basically used to knock out a gene or remove a gene or knock in or insert genes. So these are basic principles of it. Now we will discuss about RNA interference, microRNA or miRNA, and small interfering RNA. MicroRNA or it is basically small non-coding RNA that is produced naturally. It forms hairpin structures in the cell and it binds to mRNA. It binds to mRNA and block translation or causes degradation. Loose pairing of miRNA. Due to loose pairing of miRNA, it is used to broad targeting of related mRNAs. Clinical relevance of microRNA. Abnormal microRNA leads to, that is, malignancies, silencing tumor suppressor genes. So these we have discussed about miRNA. Now we will discuss about small interfering RNA or siRNA. It is derived from exogenous double-stranded RNA, for example, viral. It requires perfect pairing, that is highly specific targeting. It causes mRNA cleavage before translation, and it can be synthesized for gene knockdown experiments. So we have discussed about microRNA and small interfering RNA. So, and what are they and their differences? I hope you can understand it. Now we will discuss about key takeaways and important medical terms used and their meanings that are used in our lectures. First, we will discuss about colchicine. What is colchicine? It is basically drug that blocks cell division by disrupting spindle fibers. Karyotyping. Karyotyping, it is technique to study chromosome numbers and structure. Trisomy. Presence of an extra chromosome, 3 copies instead of 2. Tetrasomy, 4 copies of a chromosome. Microdeletion, small missing segment of a chromosome. Translocation, exchange of genetic material between chromosome segment. Recombinant DNA, DNA artificially created by combining genetic material. Knockout, removing a gene to study its function. Knock-in, inserting a gene into a genome, miRNA or microRNA, that is endogenous small RNA that regulates gene expression. siRNA, synthetic or viral derived RNA that silences specific genes. Now we will discuss the summary of our lecture. Karyotyping. Karyotyping basically helps detect chromosomal abnormalities using colchicine-related cells arrest. FISH or fluorescent in situ hybridization, it allows direct visualization of genetic anomalies like deletion, duplications, and translocations. Molecular cloning enables recombinant DNA production for therapeutic proteins. Gene expression modifications in mice, knock-in and knockout are vital for genetic research. RNA interference, miRNA and siRNA, it regulates gene expression with implications in cancer and experimental gene silencing. So we have discussed summary of our lecture.